Good morning, good afternoon, good evening, greetings wherever you may be. Welcome to this uh, lecture about the histology of muscle tissue. Welcome, and this will be a series of discussions that will help us to be able to get some insights on the histology of muscle tissue. In terms of what we want to achieve, by the end of this lecture, you should be able to identify the different types of muscle tissue in terms of the features of each muscle type and how they are different amongst themselves. And then with that, we want you to be able to describe the structural basis of muscle contraction, which muscle components help in muscle contraction. And how does that take place? You should also be able to know the elements that get to be utilized for muscle contraction. How, what, and where does this muscle contraction take place in the muscle? Eventually, we want you to understand the function and organization of muscle, how it is organized, and how that is a function of connective tissue. Then you should be able to familiarize yourself with the fact that muscle is able to regenerate. Quickly, we begin with one of the types of muscle tissue we call striated muscle, or yes, because of the regular arrangement of the units that contract in the muscle. So we start with striated muscle, which is a type of muscle categorized as striated based on the appearance, gross appearance, histologic appearance under the microscope, that it looks striated as in striped as a result of the regular arrangement of these repeated contractile components of muscles. So what are the categories of muscles that are found or that are considered to be striated or striped? These are skeletal muscle and cardiac muscle. So skeletal muscle, in terms of features, as we have planned that you need to know the features. What are the features? These are long cells and they are cylindrical in nature. Because of this, they are also referred to as muscle fibers. Fiber as in a string, because they are cylindrical and long, they are called strings or muscle fibers as well. So they also have nuclei, more than one, at least 3 or more, 4, even 4 nuclei, or nuclear cells. So these, they are found at the periphery of the cells, not in the center, at the periphery. This is as a result of the bulk of the cell, the interior of the cell, being filled with contractile elements of the cell. And contraction for skeletal muscle is typically very quick, very powerful, and it is under voluntary control. So the type of contraction is described as quick, meaning very fast to respond to stimulus, but also very strong or vigorous, under planned control or voluntary control. And these, in terms of function, we find that skeletal muscles function in movement from one point to another, what is called locomotion, and also for chewing, a physiological process called mastication, and also for phonation, as in voice projection, because the voice box is produced by sheets of muscle that make up these the voice box. Then cardiac muscle is also striated, categorized as striated muscle. And in terms of features of the cells that make up cardiac tissue, they are, yes, elongated, but they are not long. They are elongated, but they are not cylindrical in nature. They are elongated, but they are not cylindrical in nature. But they are branched. They are branched with a single centrally located nucleus. What is also an important feature about cardiac tissue is that the cells connect one to the next by a structure that is called intercalated disc. And within this structure, there are provisions, cellular provisions for intercellular communication that allow these cells to attach in that junction. So there are adhesion molecules, and also there are provisions for sharing of cytoplasm. So there are gap junctions amongst cells. The contraction for these types of cells is described as, yes, strong, but rhythmic. So the rate of contraction and relaxation is consistent. It is a rhythm. Contract, relax. Contract, relax. Lap, dap. Lap, dap. And the control in terms of this consistent rhythm of contraction and relaxation is involuntary. It is not planned, but involuntary. Then we look at the non-striated muscle type. This non-striated muscle type, we have a type of muscle belonging to non-striated muscle types called smooth muscles. Smooth because they are associated with internal organs, and in terms of features, yes, they have contractile machinery or units, but these are not regularly arranged in repeating units. Because they are not regularly arranged in repeating units, they don't show the repeated striations as a result. Therefore, they are considered as non-striated. And the cells that make up these smooth muscle tissue, they are not elongated strings or cylindrical or branched, but they are said to be fusiform or cylindrical, where they bulge in the middle and taper at the ends. And in terms of the nature of the contractions as they function, is that the contraction is slow, slower than cardiac, slower than skeletal. And this, because of the mechanism of how they produce the contraction is longer lasting. In terms of control, it is not voluntary, therefore it is not planned. Therefore, the control is involuntary. We continue in terms of muscle regeneration and growth. Skeletal muscles, they increase in size, and the increase in size of muscle tissue is called hypertrophy. The increase in the number of cells tends to help in replacing the dead cells and also for regeneration purposes. The cells that are useful for these are called satellite cells, which are proposed as the source of regeneration of muscle tissue. Smooth muscle cells also tend to increase in size under continuous stimulation, and that is also called hypertrophy. Increase in the number of cells leading to proliferation also helps in regeneration, and these smooth muscle cells tend, yes, to be proliferative. For example, the uterine or uterus, the vascular or blood vessel smooth muscles. So in there, the regenerative cells that we have are called pericytes. These pericytes, they provide a source for smooth muscle regeneration. Remember, skeletal muscle regeneration from satellite cells, smooth muscle regeneration from pericytes. The heart muscle also can increase in size, and as a result of increase in the size of the cells, this is called hypertrophy. So these were formerly thought to be non-proliferative, like they cannot regenerate. But it has been observed that after dead tissue or infarction of cardiac contractile tissue, there is a remodeling by fibroblasts. The fibroblasts that produce fiber or connective tissue fiber, and this causes scarring. New evidence also suggests that there is mitotic cardiomyocytes, and there is regeneration by blood and vascular-derived stem cells that help to regenerate heart tissue. So we begin with skeletal muscle, and so if we did a cross-section, we will be able to see that skeletal muscle tissue is very, very organized in a very interesting kind of structure. And so the basic component of the muscle tissue skeletal is the myocytes, or the skeletal muscle cell, or the muscle fiber. So that is covered with connective tissue to separate one from the other by perimysium or sarcolemma. And then we also have now these cells bundled in what is called a fascicle. A muscle fascicle is a bundle of muscle cells or muscle fibers. These are organized in a fascicle by a perimysium. So the muscle fiber is covered by endomysium. Then the fascicle is as a result of the covering of muscle fibers in a group by perimysium. And then the entire organ, made up of fascicles, different fascicles, is covered by epimysium. So this will be a skeletal muscle as seen under a light microscope. We can see the striations or the stripes. And if you remember, it is as a result of the organized repeated arrangement of contractile units. Then, yes, all these contractile units of striated muscle, what we see is that if we just get a muscle fiber, in a muscle fiber we'll find it is also a bundle of myofibrils. So a muscle fiber is also a bundle of several myofibrils. So the fibrils, each one of the fibrils is just made up of repeated arrangements of actin and myosin. The repeated arrangement of actin myosin is what makes up what is called a sarcomere. A sarcomere is the contractile unit. So the repeated arrangement of sarcomeres is what gives the skeletal muscle the straight appearance. which is here. So you have the center where the myosin, the actin are anchored, and you have the Z lines where the you have the Z lines where the actin are anchored, and you have the center where the myosin are anchored. So basically this is what we are talking about, and you can see in the top there that contraction is the actin moving towards the center, being pulled by the structure that is anchored in the center, which is the myosin. And so the coming together of two Z lines is what causes the shortening, and each shortening in a myosofibril will cause shortening of the muscle fiber, and the shortening of the muscle fibers will cause the shortening of the fascicles, and the shortening of fascicles is really what we call muscle organ contraction. So, but then the question is, how is this taking place? Basically, what happens is that the skeletal muscle is stimulated by a motor neuron electrically, and the depolarization causes the release of calcium. Then calcium that is released from the sarcoplasmic reticulum, or the cisterns or the storage sites, will be released in the cytoplasm, where it will go and bind to troponin. Troponin then will activate tropomyosin to expose on the actin where the myosin should bind, such that when the myosin now binds to the actin, then they are activated to move the actin towards the center from both ends of the Z line. This is what causes what? Muscle shortening. So in other words, we are saying without calcium there will be no activation of troponin. Without troponin there will be no activation of tropomyosin. Therefore, calcium, troponin, and tropomyosin are the control molecules in skeletal muscle contraction. That is very, very important for you to remember. So that is why we say calcium is important for coupling or linking muscle activation or depolarization to contraction, what we call excitation contraction coupling.